Good day, I am Dr. Apalata, and I decided to make this quick video because as you are aware, the cases of COVID-19 are exponentially increasing, and the situation is much more critical. And here in South Africa, the second wave of COVID-19 is already well established. So in addition to that, we have a new variant of the virus that is causing this disease that has been identified here in South Africa. And on Friday, the National Minister of Health had a press conference where he addressed the nation about the new variant. So what we are going to do is just to try to understand more what is happening with the new variant of the virus that is causing COVID-19. So I decided to title this presentation COVID-19 Revenge. But we are going to look at a number of facts to see whether it is truly a revenge or not. So we will look at the new SARS-CoV-2 variant that is identified in South Africa, and that will be the basis for our discussion. So in order to respond to the question whether we are dealing with a revenge or a viral revenge or not, we will first try to understand what is a new variant. They are talking about the variant, the new variant, and how is it different from a new strain of the virus? Is it a new strain, a new variant, the same thing? We will try to understand that. And why is it that this new variant is called 501V2 variant? We will also try to clarify that. And most importantly, we will try to look at the implications of this new variant in terms of transmission of COVID, severity of the disease, clinical presentation of our patients, in terms of infection prevention and control measures, if we have to do things differently or not, and more importantly, the effect on the upcoming COVID-19 vaccine. Is there any effect that is known or an impact on the COVID response, on the vaccine response? So before responding to these questions, what is it that we already know about the virus that is causing COVID-19 disease? And here you are seeing it's the structure of the virus. This is the structure of the virus. This is how this virus looks like. And what we can remember here, if there is something that we can remember, is that this virus has 4 structural proteins. They are 4. So the most important that we are going to have for all our discussion today is the S protein that we call the spike protein. It's a glycoprotein, and we are going to discuss this glycoprotein. It's the most critical. We will see why. But we also have the E, that is envelope proteins. We have the matrix protein or membrane protein, membrane or matrix. And we have N protein, that is a nucleocapsid. So this virus is a single-stranded RNA virus. So you can see there, it's a RNA virus. Now, the most important thing about RNA viruses is that they are able to develop mutations much more often. And that's the problem. During their replications, there are a lot of errors that happen. And one of the major problems with these viruses is that they can mutate, much more easily. And all those proteins that you see that are there, their structures and the way they are, everything is regulated by a number of genes, in their genome, in the genome of the virus. There are different genes, and those genes encode for all these proteins, structural proteins that we can see. And if there is a mutation, in the genetic element of this virus, then it will then affect the structure of any of these glycoprotein that you see. Now, let me just try to make this very clear to you. So what I'm trying to say is that whatever structure of the protein that you see on this virus, we spoke about 4 structural proteins, whatever that you see, its shape, the function that it plays, let's say the S or the spike proteins, it plays a major role. It's that glycoprotein that attach itself with the receptor of the host cell. It attach, it plays a role in attachment and fusion with the host cell at the receptor of the host cell for it to go into our body. And from there, it will replicate and be able to cause the disease. So what then happens is that, if there is any mutation in the gene that encodes the regulation of some of those proteins, if the mutations are relevant enough or they play a major role, then it might affect probably the shape or the function of some of those proteins. Then you will see that the function might whether becomes very weak or much more stronger. So it depends on the type of mutation that takes place. So that's actually what we need to remember. So then it's very important that we have this. Then we have to remember that we are discussing more about this spike protein. The spike protein has 2 subunits. It's the S1 and S2. So S1 it plays a role in the attachment, with the host cell receptors. Then the S2 will then allow the fusion to happen so that by the mechanism of endocytosis, the virus will then be taken by our human cells, our cells. So let's just try to see this. This is what you see here. So this is the virus, okay? And this is the virus, and here we have the S proteins, spike proteins are there. And this big one is the host cells. Now the host cells has in red here specific receptors that we call angiotensin converting enzyme 2 or AC2 receptors. Those are angiotensin converting enzyme 2. So those receptors are released by a number of our host cells, like there are many of them in our respiratory system, in the lung, in the back of our throat, and you can also find them in the heart and in other parts of the body. So like in our lungs, we have the cells that are in our lung. They express those receptors, the AC2 receptors. then those receptors, when you come into contact with the virus with its spike protein bind with the AC2 receptors of the host cells. So there is attachment, then there is a fusion, so then the virus will be able to progress into the host cells. So this is actually what happened. Then what I'm saying that depending on the level of the mutation that you find, if the genes that encode for the production of the spike proteins mutate, and if mutations are strong enough or more enough to affect actually those mutations will then lead into having a spike proteins whose shape can be altered, for example, or whose function can be compromised. So it depends on the level of mutations. So this is the general understanding, if we want to know what is happening. Okay, so I will come back to that to try to see the new variant that we have identified currently in South Africa, and what is the issue with that. But let's then see also how the hosts respond to all this, because the issue of vaccine becomes very important when we also understand the immune host response. Now, what happened when you are exposed to that virus? Then there is attachment, there is fusion, the virus is taken into your body by the host, by your host cells. Then what happened after that is that the first line of defense comes into play here. Okay, that's what we call innate immune response. More specifically in the lung, because most of the infection are respiratory infections, you find that we have epithelial cells, we have alveolar macrophages, we have dendritic cells. In other parts of the body, we also have natural killer cells and so on. But those are the main components of the innate immunity. So those are the immune cells who will be mobilized to come at the site of infection, to try to fight the virus. Then what will follow is that, the dendritic cells are and the macrophages, they are able to phagocyte, to swallow those viruses and digest, try to destroy them. But they will take a piece of, a small piece of that virus that we call an antigen, and they take it to present it to the T cells. Those are part of our second line of defense that we call adaptive immunity or adaptive immune response, that is run by the T cells. So the dendritic cells and the macrophages are what we call the antigen presenting cells. So they will take that small piece of antigen, they go and present it to the T cells. Now, the T cells will then become activated. Then there will be a cascade of information that will happen and the response. For example, there will be an increased production of the pro-inflammatory cytokines like your interleukin-6, your MCP1, your MIP1, your TNF-alpha. They will be all released. Their concentration will be increased into at a systemic level, or even sometimes also at a local level. So they will then play different roles, those cytokines, and most of the effect that we see, for example, when we see people developing lung infections, they develop dyspnea, respiratory inbreeding, and they become more affected because their lung cells are being destroyed by the effect of all these pro-inflammatory cytokines that are released, especially if they are released in a huge concentration. So the severity of the disease is actually the result of our own immune system, how strong it can be mobilized. So we will have a CD4 plus and CD8 plus T cells that become also highly activated. And the reason we know that they are strongly activated because we can see the presence of CD68, CD38, CD44 that are released. Then at the end, the patient becomes extremely sick, and that's the time where the patient can be admitted in our high care or in intensive care unit because the T cells become also exhausted. That's what we call T cell exhaustion. And we know that because we see some of the factors or the biomarkers like PD-1 and the NK G2A, those are markers of T cell exhaustion that can be observed. Okay, so this is how the response happen. Our body respond to the virus. Now, let's then go back on the issue of mutations. So just to try to make you understand. So I'm trying to speak more about the spike protein. Remember the spike protein, this one, the spike protein. I'm more discussing the spike protein because that is what attached to the AC2 of the host, of our cells. It's what attached to the receptor that are released by our cells. It's called angiotensin converting 2. They attach there. Now, being a protein, this protein, spike protein, is made of 3 small peptides. Now, a peptide is made of different amino acids. Like those are the amino acids, They range, those are different amino acids. And those amino acids are mainly, we have 4 bases. We have adenine, cytosine, guanine, and uracil. So each protein have a special arrangement, special sequence of those amino acids, the way they are sequenced, the way they are aligned. They are very, very specific to give each proteins. Okay, now an example of one of the first mutation, it's not the first time that we have a new variant in that we are seeing now. The new variant was also for COVID-19 was also reported in the UK, in Australia. And we are now reporting another variant here in South Africa. But this variant, for example, the D614 G mutation was initially reported, in Australia. And what was found is that there was a mutation. In that mutation, the A, that is the adenine, was replaced by the guanine, where you see that the adenine is replaced by the guanine at the position 614 of that protein, at the position 614. That's where the replacement took place. Like this new variant, it's at the position 501 where we have a mutation. So those mutations happened, with the virus. The mutation, especially the RNA virus, mutations happen much more frequently. So the virus, but this most worrying situation for the mutation that are reported in South Africa with the new variant is that there is an increase in the frequency, because the rate of the mutation, usually you have, it happens at a very slow rate. You can have around 2 mutations per month. But now scientists are seeing about 10 to 20 mutations in these new variants. So they are mutating much more often. And this situation was not seen previously, even when we had the first wave of the pandemic. it was not observed, because there are scientists who continuously do genomic surveillance to try to find out if what is happening with if there are any new mutation. But until end September in South Africa, it was not observed. But then from October and so on until now, they enrolled they look at a number of samples. They found that we have now these new problems. So some of the mutation they are observed, but most of them are irrelevant. But one, that is the N501Y mutation, that is found in the South African lineage, is highly important. It's highly important. It's almost like what was recently reported in UK that was identified. So that mutation is much more relevant, you know, but the extent at which it can affect either the structure of the spike protein or its function is not well identified. So it is not well known, you know, on the implications of these mutations on the structure and the function of the protein. So I will explain that. Let's look at the implications. Now, the first one, let's look at the implication in terms of transmission and severity of the disease. You know, so now that we have this new variant that is reported, now, is there any impact in terms of transmission and severity of COVID-19? I think that's what people want to know, you know. So this particular virus has 3 mutations on the receptor binding domain, you know. So on the spike, because that's where there is on the spike protein, that's where you find the receptor binding domain, you know, of the virus. So which is the actual part of the virus that attaches to the human cell. Okay, so that's the first thing. Now, what does it mean? There are a number of things that it can mean, although it's not yet well established. You know, there are still many studies that are going on right now, you know, by the group of scientists who are working on genomic research to try to identify this, you know. But one of the interpretation of these changes is that it is able to increase the affinity, or at least their preliminary data. According to the reports, their preliminary data showed that it might increase the affinity for the AC2 receptors. Now, so what does it mean? It means that if the virus, you know, the mutations lead to the receptor binding domain of the virus to increase its affinity to attach with the host cells, then it means that it will become, the transmission rate will become very high. It means that it will make the virus to it will make the transmission of the virus to become much more higher than what we have seen before. So what does it mean if the transmission becomes much more effective, much more frequent, much more higher than what we have seen before? It means that people can transmit the virus to each other much more easily. Okay, so it means that there is a possibility that the second wave of this pandemic becomes much more complicated. So many people can become more infected and affected than what we have seen during the first wave of this pandemic. You know, so it means that the hospital will not have enough bed. The ICUs, the high care, the casualties, we will not have enough bed, enough space to accommodate all these patients if we don't take the necessary infection prevention and control measures. So many people will be exposed to the virus. You know, if it indeed confirmed that there is an increase in the affinity of this virus with the human receptors. Now, the second thing is that supports this is that although it's still at a very early stage, to make conclusions, but at least preliminary data suggest that the virus that is now dominating in the second wave is spreading faster than what was seen on the first wave. So this is really a challenge. This is a challenge, this is a big challenge because now most of the cases, the patients that they are looking at now, most of the samples that have been analyzed and sequenced, from those samples, it's showing that the vast majority of those patients are infected with the new variant. They are infected with the new variant. So it is high likely that the second wave will be actually driven by the new variant of this virus. And since it is hypothesized that the transmission rate will be much more faster and much more easier with this new variant because of the increased affinity of the spike glycoprotein with the host AC2 receptors, then it is high likely that the second wave of the pandemic will be much more complicated than the first one. Okay, so this is what it means. Now, another thing is to support this is that they also found that when they analyzed the swab specimen taken from those patients, they found that the virus, there was a very high number of the virus from this specimen as compared to the number of virus, the viral load found in specimen collected during the first wave. So there is a higher viral load in the swabs from the patient infected during this second wave, and this may translate to a higher efficiency of transmission. So in terms of transmission, it is high likely that we will see more cases of people being infected with this second wave. But in terms of severity, it is not established. It is not clear at this point if the second wave has, if the disease now is much more severe than what we used to see before, or if people are dying more than what was observed before. So all those, in terms of severity, it's not yet established that the virus, whether it's more severe or not, it is not known. But the preliminary findings are showing that the transmission might be much more effective during the second wave as compared to the first wave with this new variant of the virus. So the second thing, what are the implications in terms of clinical presentation of this patient? Again, according to the report, it was established that clinicians have been clinicians who are managing those patients have been providing anecdotal evidence. So it means that it's not really truly established, but there are a lot of anecdotal evidence of a shift in the clinical epidemiological picture. During the first wave and in South Africa or across the globe, we know that adults were more affected than children. Adults, especially old age and people with multiple comorbidities, like diabetes, hypertension, obesity, and so on, they were more affected than very younger population and people without any comorbidity. But now, with these new variants, they are observing a shift in particular noting that they are seeing a larger proportion of much more younger patients, and patients with no comorbidities who are presenting with critical illnesses. So it is highly likely an indication that probably the clinical presentation is also going to be affected. So we will see more younger people being involved in this infection as compared to what was observed during the previous wave. So the evidence that has been collated therefore strongly suggests that the current second wave that we are experiencing is being driven by these new variants that we are seeing. Okay. Now, is there any impact or any implication in terms of infection prevention and control measures in place? The answer is no. So it means that we still stick with the same measures that the government and all agencies are establishing, the one they put in place. So we will still keep the use of mask, social distancing, washing hands, using sanitizers, and environmental cleaning, and so on, and keeping yourself self-isolated if you suspect anything. So all those measures, in fact, we still need to stick with them. We have to strengthen those measures because those measures are the one who will still continue to protect us, whether you are infected with the new variant or the old variant, it doesn't matter. So all those infection prevention and control measures are still valid, and they still need to be strengthened and strictly observed. And especially during this time where we are going through celebrations and Christmas and the New Year festivities, we really need to strictly observe the measures. We need to avoid overcrowding. We have to wear our mask properly. We need to respect social distancing. We need to wash our hands much more often using water and soap or using sanitizers. So those measures are still valid. Now, the most important question is then, are there any effects on the upcoming COVID-19 vaccine? Now, is there any way that this new variant will then make the vaccine to become irrelevant after all the efforts, after all the resources that were put in place for the development of the new vaccine? The answer is that it's not it's not established. It's not established that the vaccine will be, the effect or the impact of the vaccine will be compromised. It's not established. Although the fact that we don't know yet how those mutations all combine, the one we found at the receptor binding domain of this virus, whether they will potentially allow antibody to escape, but the full implications of the combination of these mutations still need to be studied in more details. So, but the answer for now is that there is still not any established negative impact in terms of compromising the vaccine. So the vaccine will still come in, the vaccine need to be received, the vaccine will still play a major role, even if we have this new variant. So there is no evidence as yet to show that this will negatively impact on the efficacy of the vaccine. So we don't need to worry about this, and we still expect the vaccine. So those were some of the issues that we wanted to clarify in terms of the new variant of the SARS-CoV-2 virus that is identified. You know, even the variants that were found in UK, in Australia, the same questions were raised whether there will be any negative impact on the vaccine. And until now, all scientists agree that there is no negative impact on the efficacy of the vaccine. So we still hope and we still understand that the vaccine will still be effective, and we can't wait to receive the vaccine so that we are more immunized against this virus. But at the same time, the infection prevention and control measures and practices that are in place should be strengthened, and we have to continue with them, you know, because they will still equally protect us whether you are infected with the new variant or the previous variants. And then one more thing that I forgot is that these changes, these mutations that happened so far are not enough, are not strong enough to make these variants to become a brand new strain of the virus. It's not a new strain, you know, it's a new variant. It's not a new strain, you know. So because those changes did not strongly affect the morphology, the shape, or the function of the spike cells or the spike proteins or other glycoprotein structural or functional proteins that are found in this virus. So we are not dealing with a new strain, but we are dealing with a new variant. So keep yourself safe and strongly observe the measures, the infection prevention measures, and behave responsibly during Christmas and New Year. So goodbye. Thank you.